Hi guys, because the previous two videos that I uploaded was not bad, meaning had quite a good response and students say that they really learned a lot, I decided to upload another two more videos. So I'll just label this as the discussion 3, and there's a discussion 4. So the method would be the same. The method of teaching would be the same, whereby number 1, I will start with KBSM for the first video, then the second one will move to KSSM. Okay, so to save time, guys, I want to move a bit quicker in order to save more time. I'll be going through a bit quicker compared to the previous one, and also I don't want to repeat this part here, what you're looking at here, which I already did in the previous the very first video, Amali discussion 1. So as mentioned in the first video, these are the 11 types of questions that they can ask you. And as for unguided, okay, on the next video, guys, I'll teach you how to answer method to handle using procedure. Okay, and as you all know, Amali is 15 marks. So in order to make up the points, the fourth thing that they can ask you, in order to make up the points, they can still ask you questions from guided. Okay, so let's get right into it. Why I like to use KBSM is because, guys, experiments are always the same. I always tell students we can change the syllabus a bit here and there, but certain things remain the same. The way to answer observation, the way to answer hypotheses, the way to answer variables, guys, doesn't mean that we change the syllabus. The way we answer have to be totally different. It's all the same, and moreover, I am adjusting it to your KSSM answering technique, so it's damn relevant. I tested all sorts of methods from KSSM, then I teach KBSM, then just KSSM or just KBSM. I find the best is within a class. Let's say I have a class or a session, I teach KBSM, then the KSSM, so that you can see the whole thing more completely. Okay, without further ado, let's start. Guys, okay, this is from one of the SPM paper 3 back then. Okay, so you can see they can ask you something like this. They can ask you to slot in the material and apparatus. So material here would be the 2% sodium hydrogen carbonate as well as the hydrilla. Guys, I highly recommend that as you write sodium hydrogen carbonate, you write 2% as well. Guys, you write 2% as well. Number 1. The rest would all be the apparatus. Okay, let's use another color. Let's say I use purple. Guys, let's say I use purple. So the rest are all apparatus. The 60-watt bulb, the boiling tube, the paper clip, the retort stand. Guys, retort stand, not retard stand. Be very careful, please. Retort stand, and what else? The ruler, pembaris. Okay, but normally they don't ask apparatus and material that much anymore. Guys, I'll show you a sample later. Okay, and again, in order to save time, because you guys will be conducting the experiment, katakan apakah kesan keamatan cahaya terhadap hydrilla, the effect of light intensity on hydrilla. So you'll be counting the bubbles, and to save you time counting here, okay guys, you can just fill in 10, 12, 15 and 20. I'm more concerned about how you tackle your observation and inference. If you did watch the previous video before this, guys, I told you, when you answer observation, it is slightly different from physics and chemistry. Okay, from what I heard from my friend, some of you may know Martin, and also some of you may know Brian, who teaches chem and physics respectively. They say, for chem and physics, the observation is just a comparison. Yeah. But guys, for bio, please state the value. And after that, I would recommend you all state higher or lower, because I know some of your teachers ask you to write higher, lower, but without stating the values. Actually, observation is what you see. Kalau kamu jalankan eksperimen sebegini, oh, what I observe even before I analyze is 10 bubbles release. Guys, supposed to be like that. Okay, anyway, but no problem, we can write both. So always try to go for either the highest or the lowest value. So guys, add. Okay, so we use this. Okay, when the distance, let me enlarge a little bit more. Okay guys, when the distance between light source and hydrilla is, don't forget to underline hydrilla, is 60 cm. Okay, follow exactly the RV. The total number of bubbles, I'll just continue here, bubbles release in 5 minutes is 10. If you really want to, you can say which is this is compared to 20, this is lower, because that is higher, which is lower. Okay. At the start, I told my students, you don't even need to compare, because why? Inference is where you compare and then give a reason. Guys, you give a comparison and then you give a reason. Or it's alright to give 2 reasons even. I always tell students it's alright to give 2 reasons. Look, for observation 1, the inference, you don't have to repeat the whole thing again. Guys, I've seen students answering, they copy the whole observation in inference. Tak perlu, sebab bila pemeriksa tanda inference, they must look at your observation once, because your inference is for that particular observation. So you don't need to repeat the whole thing again. Straight away write number of bubbles release is lesser or lower also can. Okay, lesser, because light intensity is lower. Okay, light intensity is lower, comma, if you want to, you can write photosynthesis rate is lower. Okay guys, that's why there are less bubbles, because less oxygen is released. You are very safe when you write both MV and RV. Yeah, because the MV is what causes the RV to be as such. Oh, light lesser, more photosynthesis. What am I saying? Light lesser, lower rate of photosynthesis, less air bubbles produced. Okay. Now, for instance, let's say if you were to write this observation on your own, guys, katakan, let's say we take 30 cm. How would your observation be? It's just a change in value. When the distance is 30 cm, the number of air bubbles released is 20, which is higher. So if you know this, guys, you definitely know your inference too. Number of air bubbles is higher, is more also can. Why? Because light intensity is higher, leading to higher rate of photosynthesis. Guys, higher light intensity, more photosynthesis, therefore more air bubbles are released. Alright, and coming to variables, I told you back then KBSM was like this. They give you, during my time we are required to do both, but now they put this under unguided. Okay, this is under guided. So very simple, manipulated variable, just take this distance between light source. Guys, distance between light source and hydrilla. Don't forget to underline. Next, this one. Total number of air bubbles released. The 5 minutes you can keep for method to handle. Okay, total number of bubbles. But if you really want to, you can add it, no problem. Release in 5 minutes. And for constant variable, guys, you can say the type of plant. Or if you want to, you can mention, I use a slightly different one from the first video that I made. So this one can be, guys, this one I think can be the, hold on one sec. This one. This one, the volume of volume of sodium hydrogen carbonate. Or concentration also can, guys, concentration also can, because you can see here the concentration is 2%. The concentration here is 2%. So I'll stick to concentration of sodium hydrogen carbonate solution. Okay, this one. Now, for the more important part, which is method to handle the variable, it is the same as what we did previously in the previous video. The method. Guys, at the start, we always use, okay, let's switch to a green, guys, something brighter. Always use different. Guys, use different distances between light source and hydrilla, that is, okay guys, we just use what you see here, the 4 values, 30, 40, 50 and 60 cm. That is 30 cm, 40 cm, 50 cm, 60 cm. Done. Like this you can get full marks. And to answer the responding variable, normally it is measure and record, but here we say count because we cannot measure the bubble. Guys, count and record the number of bubbles released in 5 minutes. Guys, do you remember in the previous video, what did I ask you to use? Stopwatch. Using a stopwatch. Sebab 5 minit kita mesti guna jam randek. Dan akhir sekali, bagi cara untuk mengawal, pemboleh ubah dimalahkan. The way to control constant variable, guys, fix the same concentration of I keep it short, sodium hydrogen carbonate solution, that is, okay, quick check, that is 2%, right? Yeah, 2%. Always double check what you're writing, that is 2%. Guys, that is 2%. Done. So this is how you tackle method to handle variable in unguided, and that is the reason why in the previous lesson I also told you that I prefer it to be unguided in a way. Next, hypothesis. Hypothesis here, you can straight away say that the higher the light intensity, the higher the rate of photosynthesis. When it comes to this type of hypothesis, the previous video I taught you there are 3, right? If you do not know what that 3 is, you can watch the very first video. When it comes to the first one, whereby there's a trend, more light, higher rate of photosynthesis, this type, right, actually got 2 types of hypothesis. One is whereby I call it a concept. Contohnya ini, concept. The higher the light intensity, the higher the rate of photosynthesis. There's another one whereby it's based on the experiment, whereby, guys, just have a look. when the distance between light source and hydrilla is shorter, the total number of air bubbles released in 5 minutes is higher. Okay, the distance shorter, more bubbles are released in 5 minutes. So be very clear and don't jumble it up, whereby one you write light intensity, another one you write number of air bubble. Guys, kelentung already, the whole thing is not good anymore. Okay? Of course, in your KSSM, this is 1 mark lah. In your KSSM, the questions are normally 1 mark or 2 marks. Of course, value will be much higher, graph is a bit higher. Okay? So guys, I'm not going to spend time to do the table and the graph here because the graph is a bit out of place lah. Yeah, and I think line graph students are quite okay. Just that again, the same advice as previously. When you draw graph, most important is to make sure that you don't use less than half the graph paper, whether vertically or horizontally. If you only use half, means that you could double your scale. Okay, you could double your scale. For every 2 cm, let's say 10 ml, you can make it 20 ml, so that it would be something like this. Example. Okay. But looking at the graph, what would you expect the type of graph to be here? Because they ask you to draw a graph whereby they want you to plot total number of air bubbles against light intensity. So the light intensity is here, then number of air bubbles is here. So understood, you will get something like this, increasing. So based on the graph, okay, this is a relationship question. So why do they ask us relationship question if it is quite simple? I find I can score quite easily. Reason is quite simple. Sometimes they want to try to catch you. Because you know, sometimes students are anxious during paper 3. You have to handle the experiment. You have to, like last year, they were using Bunsen burner. I told them, I told the first batch of KSSM students, tahun pertama baru habis MCO, saya percaya bahawa KPM takkan tanya Amali di mana mereka akan benarkan kamu guna Bunsen burner. Scared you burn something down. But guys, it has been 2 years since, and 2023 they asked energy value. Because during MCO there were many apparatus that were not in use and therefore not in good condition. So it wasn't a wise choice if they asked energy value. Okay, so coming back to my point. Because you're anxious, sometimes they just want to catch you making mistakes. Same goes to paper 2, guys. Same goes to paper 2-1. Yeah. Actually, paper 2, just to share, just in case you all are keen to know, Mr. Khor, what are you doing now? I am going to revise Form 5 in December, because earlier I revised Form 4 already. Then January, I will be discussing Teknik Menjawab, SPM analysis, topically what's important and what is not. Yeah. Okay, look at this. So the most important thing is to use back what they give you here. Therefore, the higher, okay, when light intensity is higher, the total number of air bubbles is higher. Okay, this is simply because rate of photosynthesis is higher, comma, more oxygen gas is released. Yeah. H. The experiment is repeated using 2 sprigs of hydrilla. Predict the total number of bubbles released by these 2 sprigs of hydrilla. Explain your prediction. Actually here, you need the table already. you need the table already. You give me a sec to prepare a simple table. I think I'll just do it now, real quick. Okay, so let me just remove this. A simple one, guys, a simple one, because normally table is not a problem, but sometimes they would have provided you with the table already. Okay, katakan, let's say you have the distance here, total number of bubbles, light intensity. So since we only have a few data, we can settle everything in 1 minute. Of course, the unit don't forget to put on top. And air bubbles will never have unit, so you just keep it as such. The light intensity here is 0.017, here 0.020, 0.025. Sometimes it takes a while to cool down and then continue. Sometimes. Okay, done. Okay, so 0.20, 0.25, 0.033. Okay, 0.0033, and the unit is per cm. The unit is per cm. So therefore, now they ask that the experiment is repeated using 2 hydrilla instead of 1. So how's the total number of air bubbles? Of course, there are more, correct? Of course, there are more. So we say that the total number of bubbles released in 5 minutes is more, okay, better still, more than, try to use something for the data. What is a better value to use? This? Oh, actually you don't need the light intensity also, because they didn't use light intensity, but it's okay. Should we use 10 or 20? No matter what, it will be more than 10. More than 10, okay? Because more hydrilla leads to higher rate of photosynthesis, and more oxygen gas is released. And guys, for the final question, which is operational definition, you have to, when they say what do you deduce, when they ask what do you deduce about photosynthesis, they want operational definition. Apa mereka nak salin balik? Okay, photosynthesis is, as I mentioned previously, now they only ask 2 marks in your KSSM. Dulu 3 markah. So cikgu dulu akan ajar 1 definisi, then RV and MV. But now straight we go to RV and MV. Photosynthesis is measured by the total number of air bubbles released in 5 minutes. Okay guys, please add in by hydrilla, and is affected by the distance between light source and the hydrilla. Okay, so guys, this is how you tackle any questions that range from observation all the way to classify items. Yeah. Okay, so please watch the next video, because I want to keep this as a sample, and then the next one is another part lah. Okay guys, I'll catch you in the next one. Cheers.